Hey, welcome back to Pearson Bio Academy. If you are new to my channel, this is Fazna Imran, and this is the place where you can top up your biology knowledge with our short and simple past paper video series. Here we are going to learn about Unit 1 Molecules, Diet, Transport, and Health. And today I'm going to start a topic from 1B, which is hemoglobin structure and the oxygen dissociation curve. Through this, we are going to do past paper questions from 2022 2026. So let's get dive in. And through the past paper discussion, I will explain the main points you need to study. So the first question is coming from the Jan 2020 paper, question number 7. So this is the question. Just pause if you want, you can read it. One role of hemoglobin is to transport oxygen. The graph shows an oxygen dissociation curve of hemoglobin, and you can see the partial pressure of oxygen versus percentage saturation of hemoglobin with oxygen. The first question asks you to write the definition of partial pressure. What's meant by partial pressure? So the answer is the pressure exerted by one type of gas in a mixture, right? So it is produced by one specific gas. Like if you take the atmosphere, it's a mixture of oxygen, carbon dioxide, nitrogen, and other gases. So if I'm specifically measuring how much pressure is exerted by oxygen, that's called the partial pressure of oxygen. The next question explains how the structure of hemoglobin causes the oxygen dissociation curve of hemoglobin to have this sigmoidal shape. So that's a theory point. You should know why hemoglobin doesn't form an exact straight line graph. It doesn't gradually increase when you increase the partial pressure of oxygen. You can see a sigmoidal shape. So, what it means is at the beginning there is a steep increase, right? And after some point, you can see the percentage increase starts leveling off. Then finally, it reaches 100 percentage because no matter how much you increase the partial pressure of oxygen, it has already reached the maximum saturation level. So now, how does the structure of hemoglobin cause this? You should understand the structure and how it happens. Let's see the structure. These things you should already know from theory. I'm just refreshing. Hemoglobin has four subunits, and the first oxygen molecule binds with difficulty. So it means it's like someone joining a new classroom. And it's difficult at first. The students may find it difficult to accept you. But once this oxygen gets bound to hemoglobin, it causes some changes called a conformational change. So, conformational change means changes in the tree structure. This increases the affinity of hemoglobin for further oxygen molecules. So imagine you did something good in the classroom. Making the first friend is difficult, but after some time making more friends become easier. So this is exactly what happens in the oxygen dissociation curve. Consequently, oxygen binds more easily as the partial pressure of oxygen increases. And at high partial pressure of oxygen, hemoglobin becomes fully saturated, like in the graph. When it reaches around 10 or 11 kilopascals, you can see maximum saturation. So, additional increases have no effect on oxygen binding because there is no use. As I told you, hemoglobin only has four subunits, four spaces, so they have already become saturated. There is no point increasing the partial pressure further. Now let's put all together to write the answer. Now, if you write it as first oxygen binds slowly and further oxygen binds quickly, you will lose marks. So be careful. You need to write the first oxygen molecule binds with difficulty, whereas subsequent oxygen molecules bind more easily. Now let's move to part B. The oxygen cascade describes the difference in the partial pressure of oxygen between different blood vessels and the external atmosphere. The graph shows the partial pressure of oxygen for a person at sea level and a person at a high altitude. Here you can see that at sea level, the partial pressure in the atmosphere is much higher compared to high altitude. And then you can see the trend. In the atmosphere, it's very high, but when it reaches the alveoli, there is a reduction, then arteries, capillaries, and finally veins. By the time it reaches the veins, the partial pressure becomes much lower. The question asks suggest why the partial pressure of oxygen in the air in the alveoli is lower than in the atmosphere. I really like this question, and you need to write only one point. The first suggestion that inside the alveoli, it's not only the atmospheric air, carbon dioxide and water vapor are also present because they need to be removed. And then, once oxygen enters the alveoli, it's quickly taken up by the capillaries in order to supply body cells. So, it can also reduce the oxygen availability. And if you want an extra point, oxygen can also be used by the cells lining the alveoli because they are living cells and they also require oxygen. So, these are the 3 points you can write. Now, the next question asks explain why the partial pressure of oxygen decreases as blood flows through the arteries and into the veins. So, oxygen continuously diffuses out from the blood into body tissues. As arteries branch into smaller arterioles and capillaries, oxygen diffuses from the blood into body cells. This happens because of the partial pressure difference. The blood in capillaries is rich in oxygen, so it has a higher partial pressure compared to body cells. The body cells use oxygen for respiration, which helps maintain a lower oxygen partial pressure inside the tissues. This allows oxygen to continue diffusing down its gradient. As oxygen leaves the blood, the partial pressure decreases. At the same time, carbon dioxide produced by the tissues diffuses into the blood. Therefore, by the time the blood reaches the veins, the partial pressure of oxygen is much lower than before. And the next part, determine the percentage saturation of hemoglobin in the blood as it leaves the lungs of the person at high altitude. Use the point marked X on the graph and the oxygen dissociation curve of hemoglobin. Right. So on the graph you can see point X and they are asking for the percentage saturation. So first you need to know the partial pressure here in the alveoli. The partial pressure is something like 6.2. Then you need to go to the first graph. So if you take around 6.2, then you can see from the first graph that it's around 80% saturated. So that's how you do it. You have to utilize both graphs. The next question, explain why the percentage saturation of hemoglobin in the person at high altitude is much lower than in a person at sea level. This is a very important question, and I would love to explain this because this type of question have been repeated often in past papers. Anyway, there is another question about yak animals. We will see that in Jan 2023 question 7C. Again, it's the same concept but with a different situation. So, if you understand this question, then writing that one becomes very easy. So, first, think about a person at a high altitude and a person at a sea level. Why is the hemoglobin saturation higher at sea level? If you don't know anything, just start by referring to the graph. If I go back to the graph, you can see that the partial pressure in the atmosphere, the starting point is much higher at sea level compared to high altitude. You can see at sea level it's around 20, while at high altitude it's something like 11. So this can be the first reason, right? It means that at sea level there is more oxygen available compared to high altitude. Therefore, in the alveoli also, the partial pressure of oxygen will be lower at high altitude because the atmosphere itself has a lower partial pressure. And because of that, the concentration gradient between the alveoli and the blood becomes lower. Now I'm talking about inside the body. Because the concentration gradient is lower, less oxygen diffuses from the alveoli into the blood. Consequently, fewer oxygen molecules bind to hemoglobin. That's why the percentage saturation of hemoglobin with oxygen is lower at high altitude than at sea level. And this concept was tested again in the October 2022 paper, but in a slightly different way. The question asks you to complete the graph showing the changes in the partial pressure of oxygen as blood flows back to the lungs, through the lungs, and away from the lungs. And one common mistake is confusing partial pressure of oxygen with systolic blood pressure. They are completely different, so make sure you are thinking only about the amount of oxygen in the blood. First, let's look at the blood flowing back to the lungs. This blood has already delivered most of its oxygen to the body tissues. So its partial pressure of oxygen must be very low. And it has been marked already in your graph. Some students draw it as a horizontal line to represent that partial pressure remain constant, and while others draw a very slight decrease. According to the examiner, both answers were accepted, so either is correct. Next, as the blood flows through the lungs, oxygen diffuses from the alveoli into the blood. As a result, the partial pressure of oxygen gradually increases until the blood becomes fully oxygenated. And finally, the blood flows away from the lungs. At this stage, the blood is highly oxygenated, so the graph stays high. Again, the examiner accepted two possible answers here as well. You could draw it as a horizontal line, or you could show a very slight decrease. Both were accepted in the mark scheme. The Jan 2021 paper, question 4b. Again, it starts from a graph. The graph shows the oxygen dissociation curve of adult and fetal hemoglobin. Adult hemoglobin and fetal hemoglobin, both of them have four subunits, but the difference is adult hemoglobin has two alpha and two beta subunits, while fetal hemoglobin has two alpha and two gamma subunits. Now, if you look at the graph, you can see that the fetal hemoglobin curve is shifted to the left compared to adult hemoglobin. So, what does that mean? It means at the same partial pressure of oxygen, fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. For example, if we take like 2 kilopascal from the graph, fetal hemoglobin shows a higher percentage saturation compared to adult hemoglobin at the same point. And this is very important for oxygen transportation across the placenta from mother to the baby without mixing the blood. And in the placenta, oxygen moves from the mother's blood to the fetus. At that point, the partial pressure is relatively low. So, at let's say 2 kilopascal, oxygen is limited in the environment. So, at this point, adult hemoglobin is less saturated compared to fetal hemoglobin. because it has lower affinity at this much of lower partial pressure compared to the fetal hemoglobin. So it releases oxygen more easily because it can hold only less saturated. And the fetal hemoglobin at the same partial pressure, it might still be around like highly saturated, like 50 percentage. So it holds onto oxygen more strongly. So, under the same condition, adult hemoglobin releases oxygen while fetal hemoglobin catch it more effectively. So, this is exactly how oxygen transfer happens across the placenta. Now, back to the question, explain why the oxygen dissociation curve of adult hemoglobin is different from fetal hemoglobin. So, whenever it says explain, you need both the difference and the reason. So, first, the difference. The curve for fetal hemoglobin is shifted to the left compared with adult hemoglobin. Now, the reason, oxygen must diffuse from the mother's blood across the placenta into the fetal blood. For this to happen efficiently, fetal hemoglobin must have a higher affinity for oxygen than adult hemoglobin. As a result, at the same partial pressure of oxygen, fetal hemoglobin becomes more saturated than adult hemoglobin. That's the key point. So you have to use this word same environment, different affinity, different saturation levels, which makes this to happen. This concept is tested again in the October 2024 paper. In the previous question, Pearson asked about the oxygen dissociation curves of fetal and adult hemoglobin. Now they are testing the same concept but in a different way. That's exactly why I always tell you, don't just memorize answers, understand the biology behind them. The question starts with a simple fact. During human development, the main oxygen carrying molecule is fetal hemoglobin. Towards the end of fetal development, fetal hemoglobin is gradually replaced by adult hemoglobin. And let's start with the calculation. The question says that 1 cubic millimeter of blood contains 5.3 million red blood cells, and each red blood cell contains 270 million hemoglobin molecules. The question asks you to calculate the total number of hemoglobin molecules in 1 cubic millimeter of blood, expressing your answer in standard form. So first write both values in standard form. So 5.3 million is equals to 5.3 times 10 to the power 6. 270 million is equals to 270 times 10 to the power 6. Now multiply them together. 5.3 times 270 and it's 1431, and multiply the powers, and then it will be 10 to the power 12. So the answer becomes 1431 times 10 to the power 12. Finally, convert it into standard form, so it will be 1.43 times 10 to the power 15 hemoglobin molecules. And the next question is about the structure of hemoglobin. I'm not covering that in this video because I'll make a separate video explaining every past paper question related to the structure of hemoglobin. And now let's move on to the graph. This graph shows the changes in the alpha, beta, and gamma subunits during development. This is quite an interesting graph because it's a relatively new style of question. So there is a good chance Pearson could ask us something similar again. And the question asks, describe the changes in these types of subunits during each stage of development. It's only a 3-mark question, so think logically. There are 3 subunits, so you should aim to write 1 marking point for each subunit. Also, instead of describing the graph using the month, shown on the x-axis, it's much better to relate your answer to the stages of development: embryonic development, fetal development, and after birth. Here is the model answer. So during embryonic development, the alpha subunit increases and then remains almost constant throughout fetal development and after birth. The beta subunit continues to increase throughout embryonic and fetal development. After birth, it continues to increase slightly before eventually leveling off. The gamma subunit increases during embryonic development but decreases during fetal development. After birth, it continues to decrease before finally leveling off. That's all the examiner is looking for. So one subunit equals one marking point. And the next part, the most important question. Now let's look at the uh, question first. The oxygen affinity of fetal hemoglobin is different from that of adult hemoglobin. Explain why this difference ensure that the fetus obtains enough oxygen during development. Compare this question with the one we discussed previously. So, previously they asked you to explain why the oxygen dissociation curves are different. And this time they've taken the same concept one step further. They are asking how that difference helps the fetus obtain enough oxygen. Your answer should follow this sequence. So, first state that fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. So, it means at the same partial pressure of oxygen, maternal hemoglobin has a lower oxygen saturation and therefore releases oxygen more readily, while fetal hemoglobin has a higher oxygen saturation and binds that oxygen more readily. As a result, oxygen moves efficiently from the mother's blood into the fetal blood, and importantly, the mother's blood and the fetus blood do not mix. So, this is exactly how the fetus receives enough oxygen during development. Okay, the next question is coming from Jan 2022, question 8C. So this is all under hemoglobin structure and oxygen dissociation curve. If you want, just pause and try to do it. The red blood cells in a person with sickle cell diseases are sickle shaped and less elastic. They also have a shorter lifespan than healthy red blood cells. These sickle shaped red blood cells cannot carry as much oxygen as healthy red blood cells, and they can get stuck in capillaries. The graph shows oxygen dissociation curves for groups of people with sickle cell disease and those without the disease. The shaded areas represent the range of values for each group. Be careful, don't forget to read these facts because some answers need information directly from this para. So pause and read it properly. You can also underline the important points if that helps. Now let's get back to the graph and the question. The first question, a P50 value is the partial pressure of oxygen that results in 50% saturation of hemoglobin. Calculate the largest difference in the P50 values between a person with sickle cell disease and a person without this disease. Give your answer to an appropriate number of decimal place. So you need to be very careful here. You must present the answer exactly in the way they ask. Largest difference means you take the highest value for the sickle cell disease and the lowest value for the healthy group. So, from the graph, sickle cell disease, the highest P50 value is 7.8, and without sickle cell disease, the lowest P50 value is 4.4. Now, subtract, so the answer is 3.4. Next question. identify two conclusions other than the difference in P50 values that can be made from this graph. So, first conclusion, there is a wider range of values. It means the greater variability in people with sickle cell disease. You can see the shaded area is much wider, meaning different individuals show more variation in oxygen saturation at the same partial pressure. For example, at the same partial pressure, like 5 kilopascal, different people with sickle cell disease show noticeably different saturation levels, like some above 30 percentage and some below 25 percentage. So there is high variability. But in people without sickle cell disease, the shaded area is much narrower, meaning less variation between individuals. And the second conclusion, at all partial pressure of oxygen, people without sickle cell disease have a higher percentage saturation of hemoglobin compared to people with sickle cell disease. So the healthy group always shows higher oxygen saturation at the same partial pressure. Now, the calculation question, the lifespan of a red blood cell in a person with sickle cell disease is 11 days, and this is 9.17 percentage of the lifespan of a healthy red blood cells. So we can consume that 11 is equals to the 9.17 percentage of healthy lifespan. So healthy lifespan is equal to, first you need to divide 11 by 9.17, then times 100. So it will be 119.9 days. And if you round up, it will be 120 days. And the final question, sickle cell disease can result in death. Explain why the changes in the structure of hemoglobin and the shape of the red blood cell could result in death. So, first point, the structure of hemoglobin is changed. So, it carries and binds less oxygen than normal hemoglobin. The second point, red blood cells become sickle shaped, and as stated in the question, they can get stuck in capillaries. This reduces or blocks blood flow to the tissues. Also, because of the shape change, the surface area to volume ratio is reduced so oxygen exchange is less efficient. So overall, less oxygen reach the body cells and tissues, and then you know what you have to write for the consequences. So I always ask my students to follow this approach when writing these answers, specifically whenever you want to link with death. So first, it will reduce the amount of oxygen available for aerobic respiration, and it will push the tissues to switch to anaerobic respiration, which produces less energy. And also, if this reduced oxygen supply or blockage happens in the brain or the heart, it can cause a stroke. So it can pull a person to death. Or reduced oxygen delivery to the heart can also lead to heart damage, which also end up in death. So be careful whenever the question is about death, please try to link the heart or brain. You have to use at least one example. So I hope you now have a clear understanding of this concept through these real past paper questions. If you found this video helpful, don't forget to like, share, and subscribe to Pearson Bio Academy. It really supports the channel and helps me create more exam focused biology content. I'm looking forward to bringing you part 2 very soon. So stay tuned. And if you have any question about this topic or there is a concept you would like me to explain next, leave me a comment below. Thanks for watching and I'll see you in the next video. Happy studying.